Hello everyone and welcome to today's sponsored keynote presentation, Immuno-Oncology Application of Humanized Immune System Mice Supporting Human Myeloid and Lymphoid Cells. This webinar is a part of the 4th annual event in the Immuno-Oncology virtual event series. Today's educational web seminar is presented by LabRoots and sponsored by Taconic Biosciences, a leading provider in genetically engineered rodent models and scientific services for preclinical research. To learn more about Taconic's study-ready models, custom model generation capabilities, and colony management solutions, please visit www.taconic.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You will also see a survey to the left of your screen. Please take a moment and answer the 2 questions within the survey box. Your participation is greatly appreciated. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Dr. Laura Griffin. Dr. Griffin has been working with preclinical models for a decade and is experienced in choosing appropriate models for various research applications. Dr. Griffin obtained her PhD in food science and technology at Virginia Tech, where she focused on the mechanisms by which dietary bioactive compounds influence the onset of metabolic syndrome using preclinical models. As a postdoctoral fellow at the Plants for Human Health Institute, her research shifted to focus on the impact of the gut microbiome on the metabolism of bioactive compounds. In addition to her expertise in metabolic diseases and preclinical models, Dr. Griffin is also versed in laboratory animal diets and their usage in preclinical applications. Dr. Griffin, you may now begin your presentation.
Thank you for that kind introduction. I'd like to start out with just a brief review on the NOG mouse, which was our first super immunodeficient model. This model lacks many murine immune cells, and those that are present are typically dysfunctional. These features allow for successful engraftment of CDXs and PDXs, as well as human immune cells in the form of hematopoietic stem cells, or HSCs, or peripheral blood mononuclear cells, or PBMCs. If you recall from our previous talk, the HuPBMC NOG, or the NOG mouse humanized with PBMCs, is a T cell model suitable for short-term studies like preliminary screening endeavors. But a major shortcoming of that model is that the animals will succumb to graft versus host disease, typically 3 to 4 weeks post-engraftment. The HuNOG, or NOG mice engrafted with HSCs, are also a T cell predominant model. Their lifespan, once engrafted, can be a bit longer. The major shortcoming of the original NOG model is that, as we mentioned, it's a T cell predominant model and has therefore limited capability to study other classes of immune cells. So as such, our collaborators at the CIEA developed what we like to call the next generation NOG models, starting with NOG-EXL. So the NOG-EXL was Taconic's first model that allowed for proliferation of myeloid sublineages in addition to T cells. This model was produced by transgenetically modifying the base NOG to express human IL-3 and human granulocyte macrophage colony stimulating factor, or HGM-CSF. These modifications allow for engraftment of HSCs and expansion of both myeloid and lymphoid cells. I'll call your attention to the figures on the left side of this page. You can see that when NOG-EXL mice are engrafted with HSCs from 4 different donors, the engraftment chimerism levels are largely greater than 50%, with T cell abundance between 10 and 40%, and myeloid abundance between 5 and 20%. B cells are also strongly present in this model, but they are largely non-functional, which essentially means that they don't undergo class switching. This model is available off the shelf and grafted with HSCs, which we refer to as the HuNOG-EXL. We prepare this humanized version by first myeloablating the animals at 4 to 6 weeks of age, injecting them with CD34 positive HSCs, and performing a QC check at the 10-week post-engraftment time point. We only sell animals that pass our QC benchmark, which requires the animals to have at least 25% chimerism with respect to CD45 positive cells. QC data is provided for each cohort when an order is shipped, and we have recently expanded these reports to include not only percent human CD45 positive cells, but also percentages of human B cells, myeloid cells, monocytes, granulocytes, and NK cells. Customers may select one or multiple donors per order, and for an extra fee, the HLA-2 status. So as I mentioned on the previous slide, we perform our QC checks at 10 weeks post-engraftment, but have demonstrated that human chimerism is quite stable over time in this model. Based on an internal study looking at multiple HSC donors, you can see in the figure on the left here that nearly all animals would exceed our 25% chimerism benchmark at the 10-week post-engraftment time point. There are some instances, especially with animals engrafted with HSCs from donor 3, that would not pass our QC check, so those here, but these animals would not be made available to researchers. Additionally, from the figure on the right, you can see that the chimerism levels are relatively similar or higher in the case of donor 2 at 23 weeks post-engraftment. Now I'll show you a little bit more of the breakdown of cell types that were characterized in the peripheral blood of HuNOG EXL at 23 weeks post-engraftment as part of this internal study. I would like to note that tumors were also engrafted into this cohort as part of the study. So in this context, the majority of the cells present were either CD19 positive B cells or CD3 positive T cells, but there were also myeloid cells detected and even some NK cells. The HuNOG EXL represents a more complete reconstitution of the human immune system compared to our previous models. Here's a nice close-up I'll show of the different types of T cells that can be found in the model, with the majority of this population consisting of CD4 positive helper T cells and modest abundance of cytotoxic T cells and Tregs as well. Here's a close-up of the myeloid subpopulations found in the peripheral blood. For convenience, I've labeled them with cell names rather than markers here. And I've also presented the data as both percent CD45 positive cells, as well as the absolute cell counts. So you can see that 10 to 15% of human CD45 positive cells were identified as myeloid, with monocytes, macrophages, and MDSCs also detected. Classical monocytes demonstrated the highest and most consistent presence of this cell type, but intermediate and non-classical monocytes are also present in this model. Finally, I'll share the data we have from spleens at 23 weeks post-engraftment from this study. Again, you can see, per the figure on the left, a high degree of chimerism, as well as an abundance of T and B cells and myeloid cells in the model. The myeloid presence actually was slightly higher compared to peripheral blood here. So now that we've gone through some of the basic details about this model, I'd like to show you how NOG EXL compares with the original NOG model when both are engrafted with HSCs. In this first case study, which compares the animals engrafted by a third party, you can see that the NOG EXL mice, represented in the figure on the right, consistently demonstrate higher rates of engraftment, with very few animals failing the typical QC benchmark of 25% human cells in the blood compared to a humanized NOG. Chimerism rates were also more consistent and stable over time compared to the humanized NOG mice. Although different donors were used for NOG and NOG EXL engraftment in this study, the inter-donor variability was much greater for humanized NOG mice. Donor 2 showed relatively high rates of chimerism, while donors 1 and 5 showed much lower rates. Conversely, for the humanized NOG EXL mice, the average chimerism percentages all fell within 50 to 75%. Moreover, intra-variability is much lower for the NOG EXL, with individual donor data points clustering much closer together than for the humanized NOG. Here is a close-up of the CD33 positive myeloid cells, also measured in the same study, where there is essentially little to no presence in the humanized NOG and a very consistent presence in HuNOG EXL with an abundance ranging from 5 to 20%. I'll share with you one additional case study comparing NOG and NOG EXL. So in this study, these mice were humanized and implanted with tumors from either ovarian cancer or breast cancer cell lines. You can see that the models respond differently to different tumor models based on the top panel figures. The SKOV3 ovarian cancer cell line showed similar growth for both of the models, while the OVCAR5 model grew significantly larger in the NOG, and the opposite was seen for BC038. So immune cell infiltration in the tumors was fairly similar between the two strains, with T cells being slightly more prominent in OVCAR5 in the humanized NOG, being the major differentiator there. Now I'll show you some case studies demonstrating the breadth of applications that HuNOG EXL can be used for in immuno-oncology research. So this first case study I'd like to share is one in which HuNOG EXL was used to evaluate combination therapy of a PARP inhibitor with anti-PD-1. In this study, a cohort of HuNOG EXL mice were engrafted with an aggressive breast cancer line. And as you can see in the figure I'm showing, the PARP inhibitor alone was able to rapidly reduce tumor volume. This reduction was attributed to an influx of cytotoxic and helper T cells into the tumor microenvironment, as shown to the figures on the right. After evaluating the PARP inhibitor as a monotherapy, the researchers investigated whether combination with an anti-PD-1 could improve their results. The following data was once again generated with a HuNOG EXL cohort and the same breast cancer line, this time actually showing that the combination of anti-PD-1 with the PARP inhibitor resulted in greater reduction of tumor volume in this model. So overall, these findings demonstrate that HuNOG EXL can be used to study mono and combination therapies in immuno-oncology applications. The next case study I'll share with you is one of our newer use cases for HuNOG EXL, in which the model was successfully employed to study CAR-T combination therapy for treatment of solid tumors. CAR-T cell therapy has largely been successful in hematological malignancies, and the goal of this study was to determine whether this therapy could be efficacious for non-small cell lung cancer. One of the barriers to using CAR-T therapies in solid tumors is inefficient infiltration. So in this study, researchers also investigated whether microwave ablation could assist in the tumor infiltration capacity of CAR-T cells. They engrafted the HuNOG EXL mice with solid tumors and then looked at each of the therapies alone and in combination. And ultimately, they found that the combination therapy resulted in the greatest inhibition of tumor volume, suggesting that microwave ablation could potentially improve CAR-T cell infiltration into the TME. Our next case study on HuNOG EXL and tumor-associated macrophages, or TAMs, that I'd like to showcase was performed by scientists at Bolt Biotherapeutics and was presented at the Society for Immunotherapy of Cancer meeting. This study highlights the advantages of HuNOG EXL as a HIS model to study immunotherapies, which manipulate multiple cell types, including myeloid cells. Tumor-associated macrophages, or TAMs, typically support tumor progression due to immunosuppressive effects. However, preliminary work in syngeneic tumor models has shown that activation of the dectin-2 pattern recognition receptor, which is expressed on most TAMs, actually drives a potent anti-tumor immune response. So the researchers at Bolt developed a dectin-2 targeted agonistic antibody and tested whether it could beneficially activate TAMs in a HuNOG EXL model engrafted with triple-negative breast cancer. As you can see in the figures on the bottom left, the antibody therapy demonstrated significantly greater tumor inhibition compared to the T-cell targeted therapy, Pembro. Moreover, the binding of the antibody therapy was restricted to TAMs located specifically in the tumor microenvironment based on the data shown in the figures to the right. So a very targeted therapeutic. Here is another very new and exciting use case for HUNOG EXL in the study of NK cells that was also featured at the Society for Immunotherapy of Cancer meeting. Now, before I get into the details of this study, I do just want to share that there are several models in the NOG portfolio that were specifically designed to study NK cells. These models will be discussed in our upcoming webinar in April, so be sure to register for that if you'd like to learn more about these types of models. The reason I'm sharing this example with you today is to demonstrate that it may be possible to also use HUNOG EXL to study NK cells in the context of that more complete immune system, which would be a bit different from some of our other NK cell models. So more on that later. If you recall from our introductory slides, NK cells are extremely low in abundance in HUNOG EXL. We're looking at somewhere in the 1 to 2% of CD45 positive cells, depending on the donor. Nonetheless, researchers at Zumator Biologics were able to demonstrate efficacy of their NK checkpoint inhibitor therapy in this model. You can see here the NK targeted checkpoint inhibitor was able to significantly decrease tumor volume in the model by improving infiltration of both NK cells and cytotoxic T cells into the tumor microenvironment over time compared to a vehicle control. So overall, this is a really nice example of how important it is to carefully consider what types of immune cells are essential for your therapy, as well as downstream effects on other cell populations. I hope these examples collectively relay the message that you can use HUNOG EXL to study individual cell types like T cells or perhaps NK cells, but having the presence of many different cell types could influence the outcome of your study and ultimately assist in moving therapies through your preclinical pipeline faster or with greater chance of success. So before we move on, I'll share with you two final examples. So in the first webinar in our series, we touched upon the fact that super immunodeficient mouse strains are available from different vendors. And these first generation or base versions of the mouse strains are fairly similar. Differences do start to arise, however, in the second generation models due to difference in the particular cytokines added and their expression levels. For example, it is well documented that the cytokine levels in NOG EXL mice range from 35 to 80 picograms per mil, but range from 2,000 to 4,000 picograms per mil in NSG SGM3 mice. This case study in particular showcases how these differences in cytokine levels could potentially affect your study outcomes. Scientists from Roche compared NOG and NOG EXL strains with the NSG SGM3 mouse. The researchers in this study engrafted both models with HSCs from the same set of donors in their facility, so a true head-to-head comparison could be made between the models. One key finding was a difference in lifespan post-engraftment. As documented extensively in the literature, HSC engrafted NOG mice can survive for extended periods post-engraftment, up to 1 year or more. However, myeloid supportive HIS mice have shorter lifespans due to a range of outcomes, including anemia and macrophage activation syndrome. However, the NOG EXL generally supports a longer study window post-HSC engraftment compared to the NSG SGM3, and that is exactly what the Roche scientists saw in this study. The researchers in this study also noted that human immune reconstitution was higher in the NOG EXL and NSG SGM3 compared to the NOG, which was not surprising. However, focusing in on the myeloid compartment specifically, which is a key feature of second generation models, the levels of myeloid cells in NSG SGM3 was actually not significantly different from the first generation NOG model, while HUNOG EXL mice demonstrated significantly higher presence of this cell type. Additionally, when looking at approximate composition of different types of immune cells in the peripheral blood of these animals, it was observed that the profile found in NOG-EXL most closely represented the profile of a human. In this set of experiments, the researchers examined the cytokine profiles of each model. There's a lot of data here on this slide, but I'd like to first call your attention to the central figure, which I'm boxing in red, showing the levels of HGM-CSF in each of the models. You can see that the NSG-SGM3 mice, which have significantly higher levels compared to NOG-EXL, gives rise to very different cytokine profiles in the model. So specifically, NSG-SGM3 mice demonstrate significantly higher levels of cytokines that I am boxing in purple here. And these elevated cytokine levels in the model likely attribute to some of the reduced lifespan observations that have been seen in this model compared to NOG-EXL. Now, the one gray box that I have put here showcases the one cytokine that was found to be elevated in NOG-EXL compared to NSG-SGM3 in this study. HCCL22 is a Treg chemoattractant. So this data suggests that Treg functionality may be elevated in NOG-EXL compared to NSG-SGM3. Overall, from this data set, it's logical to conclude that the NSG-SGM3 model has a higher level of cytokines compared to NOG-EXL, but also potentially a shorter study window, which is just something to keep in mind when selecting what model might be best for your study. Here's an additional data set that we obtained very recently from some collaborators at the University of Pennsylvania that also compared the HuNOG-EXL with NSG-SGM3. Now, their findings also align with the initial report from Maser in that the humanized NOG-EXL mice survived longer than NSG-SGM3 mice. In fact, the NSG-SGM3 mice that were used by UPenn did not even survive long enough for this group of researchers to evaluate their therapeutic. These animals actually died before tumors could be engrafted into the model. Reduced lifespan of the cohort was attributed to macrophage activation syndrome, which was significantly higher in the NSG-SGM3s compared to NOG-EXL. Inspection of the pathology also confirmed mast cell infiltration into relevant tissues here. And so before we move on, I do just want to note that for this study, unlike the previous example, HuNOG-EXL mice were purchased from Taconic, while the NSG-SGM3 mice were humanized at the customer site, so different donors were used, but still similar results were obtained compared to the previous example. So to wrap up this section, I'd just like to summarize some key strengths and limitations of the HuNOG-EXL model. This model overcomes several limitations compared to the first generation NOG, as it allows for expansion of both myeloid and lymphoid cells, and the cell populations are relatively stable for the lifetime of the model. However, the engrafted human T cells mature in a mouse thymus, and the levels and functionality of some cell types are not reflective of what is observed in humans. Additionally, these animals are delicate and they do require specialized care and attention compared to other immunodeficient models. For these reasons, we do highly recommend speaking with a Taconic field application scientists if you are planning to use these animals for the first time. Before we close out our discussion on HuNOG-EXL, I want to briefly talk about an access option which offers an extended study window. So if you recall the beginning of this talk, I mentioned that we engraft the HuNOG-EXL mice and perform a QC check to confirm engraftment specs after 10 weeks before shipping them to your institution. The reason we do this is to allow for immune cell reconstitution in the peripheral blood and to ensure that the animals you receive meet our chimerism benchmarks. Therefore, the soonest you'd receive a cohort of the traditional product offering, or what we call the standard access, or SA, HuNOG-EXL would be 11 to 12 weeks post-engraftment. While this may be perfectly acceptable in some cases, there could be specific instances where it may be beneficial to receive animals sooner. You might be working with a slow-growing tumor and want to engraft it as soon as possible, or perhaps you're interested in working with the model prior to the outgrowth of T cells. To accommodate these types of research inquiries, we now offer the early access or EA HUNOG EX cell model, where the animals are shipped shortly after HSC engraftment, thus maximizing your potential study window. I'll take a moment now to share some internal data that we've collected on the kinetics of immune cell populations following HSC engraftment in HUNOG EX cell. At 4 weeks, human immune reconstitution is limited, but by 10 weeks, significant human chimerism is present. Some changes to individual cell populations do still occur after that 10-week mark, such as the emergence of mature T cells, which typically occurs between 10 and 22 weeks post-engraftment. If you want a mouse with T cells already established, you might choose to order older HUNOG EXL SA animals from our on-hand inventory. If instead you want to work with mice prior to the emergence of T cells, the HUNOG EXL EA option might be a better choice. Immune cell kinetics is definitely a factor to consider when you're selecting what model might be best for your study. So while the HUNOG EXL EA option provides the longest possible study window, there are some caveats to keep in mind. Individual HSC donors demonstrate inter and intra variability in terms of engraftment. In the HUNOG EXL SA product, all animals are QC'd at 10 weeks, and any lot failures or failed individual animals are removed prior to release of animals for sale. So in this figure, the donors represented in red would ultimately fail our QC check, and no animals engrafted with HSCs from this donor would be sold. This even includes individual animals from a faulty donor that do meet our engraftment threshold. Most donors produce an average engraftment rate above the QC threshold, and these ones are the ones represented in blue. But there may still be individual animals that fail to engraft properly, which would be those that fall into the red box at the bottom of this graph. For these good donors, the individual HUNOG EXL SA animals that failed QC check would be disqualified from sale. Now, since we don't QC the early access cohorts because they ship prior to human immune reconstitution, we protect against lot failures by only using donors that have previously demonstrated successful engraftment for early access orders. While this protects against failed lots, individual animal variability will be present in HUNOG EXL EA lots, and some animals will fail to engraft. We recommend ordering additional animals per study to allow for some potential engraftment failures, as well as doing your own QC check to confirm engraftment in your animals at the appropriate time. I'd now like to share a little bit of data on the reproducibility of engraftment when HSCs from donors are used across multiple time points. So the data on the right side of the screen is from a study in which HSCs from 11 different donors were each used to engraft NOG-EXL animals on 2 different days. So you can see that in general, the engraftment rates were similar from day to day. And if a donor met the QC threshold of 25% chimerism on the first day, they also achieved it on the second day. Now, there are instances where individual animals from most donors that have low engraftment. With the HUNOG EXL SA option, those animals would be screened out. But with the HUNOG EXL EA option, those animals must be accounted for as they cannot be screened out prior to shipping, since the EA animals ship prior to a human immune reconstitution. There are several factors that can influence engraftment rates. So as I mentioned, it really is important to perform an in-house QC check if you plan on using an EA cohort to identify any animals that demonstrate low engraftment rates. your vivarium conditions, experimental manipulations, and gating strategy for flow cytometry reports can all influence engraftment rates and chimerism results. We have seen instances in which animals engrafted from the same donor, but housed at either a Taconic facility or a customer facility has yielded different chimerism results after a QC check at the respective institution. These differences were likely due to differences in husbandry and handling. However, flow cytometry protocols and percent chimerism calculations are not standardized in this field, so differing results can also be related to these factors. On this slide, I'm just showing a brief schematic of how the gating strategy for HuNOG EXL standard access cohorts are performed at Taconic. So to wrap up the discussion on HuNOG EXL standard access versus early access, I'll share one final summary slide on the models. For the standard access version of the model, you will typically receive animals quickly and can be assured that the animals have met our engraftment benchmarks. These features are good for studies in which you'd like to save on time and husbandry efforts, and also would like the option to receive animals in which T cell populations have matured before you start your study. One thing I haven't mentioned before is that you also have the flexibility to request animals at different ages in the post-engraftment process, such as right after the QC check, or maybe waiting until the 15 to 16 week post-engraftment age. Now for the early access model, that's a great choice if you need to receive the animals shortly after engraftment, and you can accept the risk of individual animal engraftment failures. It may allow you to work with slow-growing tumors and could be useful in instances where study paradigms are a bit longer. In any case, field application scientists at Taconic are always happy to help you decide which approach might be the best one for your particular study. So the HuNOG EXL is a great model because it can support both myeloid and lymphoid lineages. But as novel IO therapies targeting specific cell types have emerged, the need for models that can hone in on specific cell subtypes has increased. For this reason, additional NOG models were developed. And so for the last part of this talk, I'd like to briefly go over one additional myeloid supportive strain, the HIL6 NOG. Tumor-associated macrophages and myeloid-derived suppressor cells, or TAMs and MDSCs, are of interest due to their immunosuppressive features and are thus a target in IO research. Interleukin-6 is an important cytokine for induction of TAMs and MDSCs, and so with increasing interest in therapies involving these cell types, the HIL6 NOG, which expresses human IL-6, was designed. To the right here, I'm showing some of the original published data on this model, where comparisons were made between HSC engrafted HIL6 NOG, NOG EXL, and the original NOG strain. So the animals in this study were engrafted with HSCs, and then later on with the tumor cell line, HSC4, which is human head, neck, and squamous cell carcinoma. So back to the data presented here, you can see that in the top figures comparing the HIL6 NOG with the parent NOG strain, that CD33 and CD14 positive monocytes were more abundant at all time points in the HIL6 NOG model, and that CD66 B positive granulocytes were more abundant in the early stages post-engraftment, but leveled out after 8 weeks. Similarly, for the bottom panel figure, we see significantly higher levels of monocytes and macrophages in peripheral blood and spleen for HIL6 NOG mice, but greater abundance of granulocytes and MDSCs in humanized NOG EXL, particularly for the spleen. The HIL6 NOG mouse has been available for many years, but there has been less work done on it compared to the NOG EXL. I'll share with you some unpublished data that was provided by an anonymous pharma company that actually compared the HIL6 NOG and NOG EXL. Both of the cohorts were engrafted with HSCs at Taconic, and the researchers looked at immune cell populations after 23 weeks post-engraftment. these HSCs were engrafted from the same 3 donors. So first, let's take a look at the innate immune compartment of each model. Researchers saw that there were overall similar engraftment rates for myeloid cells, with this cell type accounting for approximately 7 to 8% of human CD45 positive cells recorded for the models. Monocyte levels were relatively similar for both, but non-classical monocytes was an exception. For the granulocyte compartment, researchers saw significantly higher engraftment in HuNOG-EXL compared to humanized HIL6 NOG due to a higher concentration of basophils, eosinophils, and neutrophils. These results align with what was observed in the original HIL6 NOG publication. So now let's take a look at the adaptive immune cell population in each of the models. The researchers actually saw that B cells were more abundant in the HIL6 model, with B cells representing around 55% of all human CD45 positive cells. In contrast, only 35% in HuNOG-EXL mice. Remember to keep in mind that these cells are largely non-functional, but still, if you'd like to have a model with higher abundance of this cell type, then perhaps the HIL6 NOG should be something to consider. I'm also showing on the right some data on effector Treg populations. For humanized HIL6 NOG, there are essentially very few Tregs present. And in contrast, for HuNOG-EXL, they account for about 5% of all CD45 positive cells. So perhaps if Tregs are important for your model, maybe the HuNOG-EXL is one that you should consider. The last piece of data from this study pertains to the NK cells in each model. We've spent a little bit of time touching on NK cells today, and as I mentioned, we do offer some models specific to studying NK cells, which will be discussed later. In this study, NK cells were found to be just a little bit higher in the HIL6 NOG compared to NOG-EXL, but overall the abundance of NK cells is very low. We're looking at about 1 to 2% of all CD45 positive cells. So NK cells might not be a primary target when thinking about these models, but it can still be useful to have them present as they could interact with the other cell types as you're adding in tumors and therapeutics. Another consideration when thinking about NOG-EXL compared with HIL6 NOG is lifespan post-engraftment. We've seen that NOG-EXL has a relatively long lifespan post-engraftment, but it does still have its limitations. And we actually have heard reports that HSC engrafted HIL6 NOG mice have extended survival. So this might be a good choice to look at for studies where you might need a very extended study window, longer than typical. Before we wrap up, I'll share with you one final case study looking at the HIL6 NOG, where tumors were engrafted into the model, which was not the case for the previous example that I shared with you. In this case study, researchers were looking to test an antibody therapy targeting specific monocyte progenitors with the intention of inducing apoptosis in proliferating monocytic progenitors without harming non-proliferating mature monocytes, ultimately looking to lead to a reduction in TAMs and tumor volume. To test the efficacy of their antibody therapy, H22 in vivo, an HIL6 NOG model was used. The cohort was engrafted with CD34 positive cells and HSC4 cells 2 months later. Panel B here shows that the human immune cells infiltrated the tumor, and in panel C and D, that the novel therapy, H22, significantly decreased monocytes in circulation and in the tumor following treatment compared to the control antibody. In addition to the reduction in monocytes observed in the previous slide, the researchers also saw reductions in TAMs without concurrent reduction in total volume of human leukocytes or non-monocytic cells, as well as a reduction in HSC4 tumor volume and weight with the novel therapy, which you can see in the bottom panels. So overall, this is a really nice example for a use case for HIL6 NOG for studies involving monocyte targeted therapies. So we've now learned a lot about the NOG-EXL and the HIL6 NOG strains, and I'll just say a few words to wrap up. As I'm sure you've gathered from our time today, there are many factors to consider when choosing a model for an immuno-oncology study. Some of these factors are related to simple logistics, and other factors are more complicated, like which immune cell types are needed and when to engraft tumors. There is no perfect model, but the scientific team at Taconic is here to help you determine which one might be best for your individual needs. We're happy to share data and support your endeavors as needed. So at this point, you've learned about the NOG mouse and a few of the next generation NOG strains. All of these models have their merits and can be used for various IO therapies. And with that, I will say thank you for your attention.
Thank you again, Dr. Laura Griffin, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Taconic Biosciences, for sponsoring today's webinar. Please visit Taconic Biosciences' booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed via the contact information you provided at the time of registration. You can also contact a Taconic scientist directly to learn more about how humanized mouse models can accelerate your drug discovery pipeline at www.taconic.com. This webcast can be viewed on demand for 1 year until April 30th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.